A 65-year-old man presents to emergency department with confusion, muscle weakness, and signs of respiratory disorder. Okay, respiratory distress. His medical history reveals infusion of high dose of thiopental. High dose of thiopental. Okay. The laboratory test shows elevated lactate levels and metabolic acidosis. Okay. Which of the following best explains by which thiopental affects this patient's condition? Okay. See here, the patient is having elevated lactate levels and metabolic acidosis. Okay. It's also having respiratory distress. Okay. If you see that this options are not so, this history is not so straightforward. Let's see the options now. Inhibition of ATP synthase in the electron transport chain. I think this question is mainly to test the knowledge on inhibitors of electron transport chain. Okay, the first option is inhibition of ATP synthase in the electron transport chain, competitive inhibition of ubiquinone at complex 1, disruption of electron transport from NADH to coenzyme Q at complex 1, inhibition of cytochrome C oxidase at complex 4. Okay, fine. Here, the other clue is here. So they're asking you about the inhibitors of electron transport chain. Now let's see whether the history shows any compounds or any substances that can be an inhibitor. Thiopental. Okay, thiopental. Is it an inhibitor of electron transport chain? Yes, thiopental is actually comes under barbiturate compounds. Okay, so those compounds inhibits complex 1. Okay, so complex 1 is NADH coenzyme Q oxidoreductase. Okay, so thiopental can inhibit complex 1. So when complex 1 is inhibited, electron transport chain will not happen. So complex 1 is inhibited. So ETC is inhibited. So ATP production will not be there, etc. Okay, so that can lead to lactic acidosis. Okay, because NADH is not getting oxidized because NADH enters in complex 1. Okay, so NADH is not getting oxidized. Okay, so NADH gets accumulated, which converts pyruvate to lactate that can cause lactic acidosis, which is nothing but metabolic acidosis. Whenever there is metabolic acidosis, what happens? There will be respiratory compensation. So the patients will have respiratory distress. So that is the answer. Okay, this patient is likely to have barbiturate poisoning or thiopental overdose, which is known to inhibit complex 1 of electron transport chain. Okay, so the answer here is disruption of electron transfer from NADH to coenzyme Q at complex 1 of electron transport chain. That's it. Okay, this is slightly a difficult question, but if you know the basic concepts, it will be easy. Okay, just to give you a snapshot of what is electron transport chain. So electron transport chain happens in the intermembrane space. Okay, imagine this is a outer mitochondrial membrane. This is the inner mitochondrial membrane, and this is the matrix. Okay, and all the substances responsible for electron transport chain are present in the inner mitochondrial membrane. Complex 1, complex 2, complex 3, complex 4. Okay, there's something called ubiquinone, which is also called as coenzyme Q. Okay, Q is also called as ubiquinone. Okay, so NADH, first point, NADH enters into complex 1. This is complex 1, this is complex 2, this is complex 3, this is complex 4. Okay, during this NADH will get oxidized, 4 protons will be pumped into the space, and the electrons will be generated that will move from here to coenzyme Q, and from here enzyme will enter into here, complex 3, and from there it goes to complex 4. This is how electrons will get transferred from complex 1 to 3 to 4. Okay. See, complex 3 is cytochrome C, coenzyme Q, oxidative reductase. Complex 4 is cytochrome C oxidase. Here is what oxygen will be reduced to form water. This is the final step. Okay, this is how NADH is transferred to electrons and gets oxidized. Okay, during the electron transfer in the complex 4, protons will be pumped into the space. In the complex 3, 4 protons will be pumped into the space. In complex 4, only 2 protons will be pumped into the space. So this whole intermembrane space will be full of protons. Okay, from complex 4, it enters directly into coenzyme Q, and from there, electron will be changed. Complex 2 will get its electrons and protons from FADH. That's it. Okay, so this is what happens in the electron transport chain. Okay, as electron gets transferred from complexes, protons will be pumped into the membrane space, and there will be protons sitting in the space. Okay, and these protons will generate, all these protons will generate something called proton motive force. Okay, and this proton motive force will act on the fifth complex, or there's one enzyme called ATP synthase. ATP synthase. Okay, so this proton motive force, these protons will enter through this ATP synthase. When this enters into the ATP synthase will get activated, which converts ADP to form ATP. That's it. That is how ATP is produced. Okay, so that is how NADH will produce. So that is how ATP is produced. Okay, that is how NADH will produce 2.5 ATPs and FADH will produce 1.5 ATPs. That's it. So the electrons will move from one complex to other complexes, generating a proton motive force. And this proton motive force will act on the ATP synthase complex and produces ATP. Okay. So NADH produces 2.5 and FADH produces 1.5. Okay. There are inhibitors targeting each of these complexes. Complex 1 is targeted by rotenone namobarbital. It's nothing but a barbiturate compounds. For example, this patient we have given thiopental is nothing but a barbiturate compound. Okay. Complex 2 is inhibited by malonate. Okay, that's it. Complex 3, antimycin A, dimer-caprol, and British anti-levisate that can inhibit complex 3. Complex 4, cyanide, hydrogen sulfide, carbon monoxide. These are complex 4. Okay, fine. So when any of these complexes are inhibited, what happens? Electron transport chain is inhibited. So what happens? ATP is not produced. Then what happens? NADH will accumulate. What that will do? That will cause lactic acidosis. What that will cause metabolic acidosis. What happens when there is metabolic acidosis? There will be respiratory compensation. Okay, to reduce the carbon dioxide. So there will be hyperventilation. So they'll have respiratory distress. That's it. Okay, fine. So that's about this electron transport chain inhibitors. Okay, fine. So this is very, very important. Okay, so when it comes to the last complex, ATP synthase, okay, for it to function properly, ADP should come into the matrix. Okay, for that we need something called ADP ATP translocase. Translocation should happen. Okay, then this ATP synthase consists of two subunits, F0 subunit and F1 subunit. Okay, we'll see about this in the next question. Okay, fine. The next question is, a researcher is studying the effects of various inhibitors on oxidative phosphorylation. Okay, in the mitochondria. Okay, a researcher has isolated the mitochondria and is putting various inhibitors and seeing how oxidative phosphorylation changes when he puts each of this inhibitor. Okay, when a specific inhibitor is added, oxygen consumption decreases and ATP production stops. Okay, oxygen consumption is decreased, ATP production is stopped, but the electron transport chain remains functional. Okay, each and every word is important here. Okay, which of the following is the most likely inhibitor? Okay, see what are the features of this inhibitor? This inhibitor is added. Let us name this inhibitor as inhibitor X. When this inhibitor is added, what happens to electron transport chain? Electron transport chain is intact. That is, electrons are moving from one complexes to another complexes. You understood? Electron transport chain is inhibited. That means electrons is moving from complex 1 to complex 2, complex 3 to complex 4. That is clear. Okay, because it's intact. Okay, what are other features that has been observed? Oxygen consumption decreases. Oxygen consumption decreases. Okay, fine. The electrons are moving from 1 to 3 to 4, but ultimately the oxygen is not getting converted to water. Okay, fine. Next, ATP production stops. ATP is not produced, so ATP production is stopped. Okay, this will happen only when you inhibit some, or when you add a inhibitor that is going to target ATP synthase. Your electron transport chain is normal. Electrons are getting pumped in, protons are getting pumped into the space. Okay, now oxygen is also getting, your ATP is not getting produced, so oxygen is not getting reduced to water. Okay, so this is a clear-cut case of adding an inhibitor that is targeting the last thing, that is ATP synthase. Okay, fine. So as I told you earlier, ATP synthase has two subunits, F0 and F1. Okay, if you see this figure closely, you can see that. Okay, this is the F0 subunit and this is the F1 subunit. F0 subunit is in the present membrane and F1 will protrude into the matrix. Okay, so when the proton enters into this space, into this ATP synthase, this F0 subunit and this bent axial will rotate. Okay, F1 subunit will remain static. Okay, once this rotates, the ADP will be converted to ATP. Okay, this is how ATP is produced. Okay, but when you add an inhibitor called oligomycin, it is going to target the F0 part of ATP synthase. So oligomycin will inhibit F0 part of ATP synthase. So what happens? The F0 subunit and the bent axial will not rotate. So what happens? ATP will not be produced. When the ATP is not produced, oxygen consumption will also be stopped. Okay, even though the electron transport chain is functional, okay, only when you convert the ADP to form ATP, your oxygen consumption will increase. Okay, so this is a classical case of an inhibitor targeting the F0 part of ATP synthase. Okay, one. Okay, we already know what are other inhibitors here. Routine, that is for complex 1. Antimycin complex 3. Cyanate complex 4. That's it. Okay, if you remember carefully, I told you that for ATP synthase to happen, we need this thing to happen, ATP-ADP translocation. Okay, this is inhibited by one more drug called atractyloside. So both atractyloside and oligomycin will target ATP synthase. Oligomycin, especially F0. Okay, fine. That's about it. The answer is very clear. Okay, so first we should know what is the inhibitor doing. Inhibitor is keeping the electron transport chain intact. Okay, it is not disturbing it, but oxygen consumption is reduced and ATP is reduced. So there is a problem in ATP production, which is done by ATP synthase, which has two subunits, F0 and F1. F0 is targeted by this inhibitor called oligomycin. Okay, in this context, I want to give you a small brief over one more compound called uncouplers. Okay, what are uncouplers? Uncouplers will uncouple this oxidation from phosphorylation. Okay, how this uncouplers, if you see classically or typically, the protons which is pumped into the intermembrane space will enter through this ATP synthase and ATP is produced. That is normal thing. If we add an uncoupler to the mitochondria, what will happen? All the protons will come through these uncouplers. instead of going into the ATP synthesis, everything will come through this uncoupled channels. So what happens? Proton motive force is not converted to ATP in the presence of uncoupler because they are not going into ATP synthase. Instead, this proton motive force is generated in the form of heat because everything comes out through the alternate channel. Here, oxygen consumption will be there, okay, because all protons are coming inside, but there won't be any ATP production, and instead there will be a heat production, okay. So this uncouplers are those things. What are various uncouplers? Uncouplers can be either physiological uncouplers or synthetic uncouplers, okay. Physiological uncouplers, I will tell you: thyroxine, uncoupler protein 1 present in brown adipose tissue. Synthetic uncouplers: 2,4-dinitrophenol, FCCP, okay, that's it, okay. That's why they use uncouplers to lose weight initially in the olden days, which is now not advisable because there are a lot of deaths reported from uncoupler abuse, okay. Can lead to weight loss because all your energy is displayed in the form of heat rather than ATP, okay. Fine, that's about it. But this UCP1 protein of brown adipose tissue, UCP1 present in brown adipose tissue, is very, very important for thermoregulation in infants, okay, present in the infants, present in the interscapular region, okay, newborns, okay. So that's about this uncouplers, okay, fine. That's about this question, okay.